Good morning, everyone. I welcome you all to this 14th day of CSIR Life Science question and answer discussion session. Today, we are going to discuss about the Unit 4 cell communication and cell signaling. It's a remaining part. Past 3 days, we are discussing the part C questions of Unit 4. Before I enter into the session, please let me know, am I audible? Can you see my slides? Is it visible? Please let me know through chat. Yeah, thank you for your response. We will start the session. Okay, this is first question. A potential valuable therapeutic approach for killing tumor cells without affecting the normal cells is the use of immunotoxins. So, the potential valuable therapeutic approach is a immunotoxins using approach. While using the immunotoxins that not kill the normal cell, it's only killing the tumor cells. Immunotoxins constitute monoclonal antibodies against tumor cells, cell conjugated to lethal toxins. So, these immunotoxins constitute monoclonal antibodies against tumor cells conjugated to lethal toxins. Which of the following molecular approach to you think is not appreciable for generating tumor cell specific immunotoxins that will not kill normal cells? Okay, the cost they are provided to is the method. The immunotoxin is killing only the tumor cell. Another step, this immunotoxins constitute monoclonal antibodies against tumor cell conjugated lethal toxins. Which of the following molecular approaches do you think is not appropriate for generating the tumor cell specific immunotoxins that will not kill the normal cells? Here they are asking not appropriate, not correct method to generate the monoclonal antibody. For this technique, we want to know clearly about that immunotoxins and the monoclonal antibody. Here they are asking the method of which is the best, which is not a best method for monoclonal antibody generation. So, as per their given statement, C is a wrong one. So, first we will discuss the correct methods. Cell surface receptor binding polypeptide chains of toxins molecule which should be replaced by the monoclonal antibodies against particular tumor cells. Okay, this tumor cell surface is modified. The cell surface receptor binding polypeptide chains of toxin molecules. Okay, toxin molecules having the receptor domain. That domain is that is receptor binding polypeptide chain that is replaced by a monoclonal antibody against particular tumor cell type. Okay, if for example breast cancer, there is a HER2 is a receptor, a recognition receptor on the cell surface. So if in this method they are designing that immunotoxins with uh, immunotoxins means they want to design that molecule, that peptide chain is replaced with the monoclonal antibody against the HER2. That is, the HER2 is a human epidermal growth factor which highly present on the breast cancer cell lines. Normal cell lines also there, but specifically it's present in the breast cancer cell lines. So this is first statement, and next second statement also right. Constant region of FC domain of tumor cell specific monoclonal antibody should be replaced by the ligation of toxins. So this monoclonal antibody FC region is replaced with the toxins. These toxins are easily recognized by the our human system, so it's not attack the normal cell, it's only affect the human cell, tumor cells. Then third question, it's in this question, variable region, that is the FAB domain of tumor cell specific monoclonal antibody should be replaced by the ligation of toxins. So in this they are provided that variable region FAB. So whenever you are studying the concept about the monoclonal antibody production, we are not acceptable to change the variable regions. While you are changing the variable region, that is change the character of this monoclonal antibody, so it's not acceptable. That don't replace that particular place. Okay, we can conjugate with the FAB region, that variable region, but we won't replace that region. So this third statement is wrong for the production of monoclonal antibodies. Inhibitor polypeptide chains of toxins should be conjugated to FAB. Here they mention conjugation. Conjugation means is a binding, it's additional attachment. Okay, that is acceptable. So in this case, they won't change their character. So this all about the monoclonal antibody. So whenever we are studying a product producing the monoclonal antibody, we are not recommended to replace the FAB region. Product producing the monoclonal antibody, we are not recommended to replace the FAB region. Next thing about next question, and this question, in cells having G protein coupled receptor, inhibition of protein kinase A by siRNA technology led to diminished transcription. Androgen binding protein ABP and CREB protein. Addition of CAMP, which is the second messenger, will lead to, here they provided the statement. Additionally, they are asked, we want to understand the statement. That cell having G protein coupled receptor. This is the very predominant cell protein in the cell surface. Okay, they are if you are following the whole classes, means you can know, you can remember this, that G proteins are having these 7 transhelix in the membrane. Okay, they are having the membrane, this is the membrane protein. Said 7 sheets are there, that are they are one they are helpful for the ion intake and ion release to the external surface. So G protein coupled receptors inhibition of protein kinase A, and protein kinase A is another it's a major role playing compound present in the cell by siRNA technology. So, lead to diminished transcription of androgen binding protein and CREB protein. There's the two processes are changed with the this androgen binding protein and the CREB protein. So proteins are changed by the siRNA technology. Here the question is additional of CAMP, which is second messenger. It's lead to, if they are having the activation of cyclic AMP, there is leads to a no change in transcription level. Okay, there is a no change in transcription level. This additional cyclic AMP lead the upcoming process, so there is no change It's a water molecule soluble, water soluble hormone signaling pathway. This kind of this concept, the question number 57th concept is fully about the water soluble hormone signaling pathway. Whenever you are studying the signaling pathway, in the signaling pathway we have two different pathways. One is water soluble hormone pathway, another one is a lipid soluble hormone pathway. This water soluble hormone pathway, we are having G protein activation. After this G protein activation, that protein kinase is active, getting the protein kinase, it's helpful for activate the protein. In this pathway, cyclic AMP is additionally added as a secondary messenger. Addition of this secondary messenger, it's lead the full upcoming pathway. So there is, if they are change, if the researchers changed the this G protein with the siRNA technique changes, because they are additionally having the this cyclic AMP. This cyclic AMP is that can carry out all the transcription process. There is this one this G protein activation is only in a membrane level. Internal cell surface, internal cell liquids are having cyclic AMP, that leads a transcription. Next question is, the binding of ligand to cell surface receptor activates an intracellular signal transduction pathway through the sequential activation of 4 protein kinase. Okay, this cell surface receptor activities activates an intracellular signaling transduction pathway through the sequential activation of 4 protein kinase. In the human cell line A, here this is the statement. Here the question statement is started. In the human cell line A, these kinase are held in a signaling complex by the scaffolding protein, whereas in another cell line B, these kinase are freely diffusible. Which one of the following possibility do you think not correct? Okay, they are doing the complexing in a human cell line A. These kinase, okay, the protein kinase are held in a signaling complex by a scaffolding protein, whereas in a signaling cell line B. So cell line A and cell line B having the link that are using these scaffolding proteins. These kinases are freely diffusible. Which one of the following possibilities is not correct? Okay, here not correct is C answer. First we will discuss the correct answers. Speed of signal transduction will higher in a signal A. It's the correct one. Why? Because that signal protein kinase, this cell surface has scaffold proteins are make activating. Okay, this diffusion rate is freely diffusible. It's in a easy manner. So the signal transduction is in a high mode. Another case is a possibility of cross-linking with the another signal transduction pathway will be lesser in a cell A. Okay, there is no cross-linking. It make a scaffold and it's fused to B cell. So there is a no cross-linking within the signaling transduction pathway. Then another right answer is D, that potency of spreading signal through other signal pathway will higher in a cell B. Okay, cell B is having the scaffolding protein, so it's higher, that can easily spread the signal. So wrong answer is for this concept, that is A and B cells are going to be complex with the scaffold protein. During the scaffold protein binding, the diffusion is become easy, that protein diffusion, that protease diffusion. So kinase diffusion. So in this concept, what is the wrong possibility? Possibility of signal amplification will be higher in a signal A, that cell A. So that is, they provided the statement called possibility of signal amplification is higher. This statement is wrong. Why wrong? Signal amplification means that signal It's multiply within the one particular region. Okay, comes under the that interior and the interior signaling pathway. This kind of concept, just imagine in the amplification process, in a one step, transferred signals are multiplied, but in this case that is not multiplied in this cell. The cells complex and basal and B cell is complexed with the scaffold protein. That scaffold protein just exchange the molecule, exchange the transfer the signal. There is a no amplification process is there. Okay, next is very very important and very easiest question. Just imagine and understand the concept. Okay, uh, please imagine the whole statement. Cells and apoptosis by two distinct and interconnected pathway, that is extrinsic and intrinsic pathway. Extrinsic pathway is activated by extracellular ligand binding to cell surface death receptor. Whenever a apoptotic stimulus activities intrinsic pathway, the pro-apoptotic box and pack proteins become activated and induce the release of cytochrome C from mitochondria, leading to caspase cascade activation resulting in apoptosis. In cell A, this is the prior information for you that uh, till now we read the information. Okay, cell having two pathway, extrinsic and intrinsic. Extrinsic pathway, extracellular protein attached on the surface, that attachment activating the intrinsic pathway. Externally some compound, that may be the drug, that may be the signal, that compound attach on the extracellular receptor. That attachment makes the intrinsic pathway activation. In this intrinsic pathway activation, pro-apoptotic parks and pack proteins become activated. This is the one. This activation, it's releasing the cytochrome C, okay, from the mitochondria. It makes a releasing. After this releasing, it's leading to caspase cascade activation. Caspase cascade activation. Okay, this is the final step. This caspase cascade activation makes a apoptosis. This is the story. Here the question is is introduced by micro injection, whereas in a cell B, this cytochrome C introduced by micro injection, but box and back are inactivated. Okay, here the main important concept statement is what they ask. The injected the cytochrome C, but that is not, there is no inactivation of backs and back, that is in an inactivated mode. What will be the most appropriate apoptotic response? Okay, here the response is they both in the cell. Okay, they are injected the cytochrome C to both cells. A very very easiest and interesting easiest question. As per their statement, intrinsic pathway first activated by the extracellular signal, first point. Second point, this intrinsic pathway make a pro-apoptotic pathway proteins activation, that is the back and box. After the activation, released in the mitochondria. So third step is the mitochondria. After the mitochondria release, they are leading the cascade activity. Thus then apoptosis automatically happen. So in this step they provided, that is the story. In that story they are asking the question, cytochrome C is introduced by the micro injection. There is a no, we don't need the box and back protein for the cytochrome release. They are directly inject the cytochrome C to B cell A and cell C. Sorry, cell A and cell B. Additionally, they are given or confusion statement, that is the back and box in inactivated mode. Why we want back and back? Okay, we don't need. Okay, in this apoptosis pathway, this back and box proteins are helpful for production of cytochrome C. Here in this case, in this research study, they are directly injected the cytochrome C. Then why we need the box and pack? So this statement is a confusion statement. Just ignore this statement. As per their statement, cytochrome C is injected. After the injection, our caspase cascade become activated. This activation leads to apoptosis. So percentage of injection, percentage of the apoptosis is higher in the cell A and cell B. It's in a similar stage. So the answer become A. So whenever you are seeing this kind of question, okay, this is big paragraph, we can't able to answer. The CSIR, in one thing you want to understand, CSIR peoples are understand our ability. Okay, we are all not expert in all the field. We are expert, so they are provided this story. Okay, this first 4 lines given story about the apoptotic pathway. This apoptosis pathway are having a they are giving the better idea that leads you to understand the apoptosis concept. Then only they are providing the question. This is the helpful for the understanding. Next, in this concept, uh, like this concept, many of the questions we can expect a number of question. That question, just take a time and read the question. Okay, uh, many of the questions are direct answers, but we don't have the time in the proper time in the examination period. So we quit these some questions. There is a possible, if we are attended means you may know that. If we are new people, please listen. Once you are enter into the examination hall, just fix the time for part A, part B and part C by yourself. Okay, in this case, we are all no we are all expert in some portion, but in that portion we will spend more time. So there is a possibility to leave some questions without reading. So please understand, uh, when going enter into the examination hall, please divide your time. How we can divide the time? Okay, there is a examination period is 3 hours period to the whole 9 to 1 yeah 1 a.m. Okay, sorry, 12 actually that is a 10 to 12, 3 hours time. In that period, first 20 to 30 minutes, just divide 20 to 30 minutes you can spend for the part A. After that, just move to the part B. Don't stay on the part A questions. It's not recommended, because some engineering students may well versed in the this kind of part A questions, so they are not acceptable, no, not needed one. So don't spend more time in a part A. Then part B. Okay, part B, there is n number of questions are there. Okay, just read fastly the question, because in that part A is we are having only 2 marks. That 2 marks is sometimes that questions looks biggest one, that read. So just fix the first 1 hour for part B. Okay, there are 50 questions are there. Just understand, first 30 minutes for the your part A, then part B we want to spend the 1 hour. Why we need 1 hour? That 1 hour means that carries 70 questions. That is very important. If you are understand and read the part B well, means that part B and part C is interlinked. There are many questions, interlinked questions, so you can answer the part C with the help of part B. So that is very important thing in this CSIR examination. Whenever they are asking the part B question, you are reading the part B, read thoroughly. That's the answer for some of the part C questions are there in a part B. Part B directly they are given as a statement, they are given as a question, that will help you for the concepts in the part C. So that given just give a 1 hour period, then move to the part C question. So part C question, you want to read, thoroughly read. Okay, once read the question, then second time read the question. After that reading of second time, you will get some grasp. Okay, this is the concept. If you are not getting the concept, just move to the next question, don't stay in the first question. Okay, just remember, whenever you are enter into the part C, there are you having 1.5 hours. So 1.5 hours you have 75 questions. In that you want to answer 25 questions alone. So once read the question thoroughly, then once again read the question. If you are getting the concept, if you are getting a related answer, again read the question. Otherwise just quit the question and move to the next, don't stay in the first question. This is the just information about the part C attending question. Okay, this kind of concepts are very easiest one. Just first reading itself you will get the better understanding. One more thing for botany, zoology. Okay, I am a biotechnology student, I'm not well versed in zoology and botany, but in this kind of question, on the other hand, some of the botany and the zoology people, they are not known this apoptosis concept. So whenever you are seeing this word, okay, okay, this is mitochondria, back box protein, okay, this is, I am not relevant to this question, I'm just quit and move to my part, that is not recommended. Okay, whenever you are entering into this part C, they are provided the story. If you are reading the story, you will get understand. Whenever you are in a zoology and botany student, you can easily understand. So don't quit the question with their keywords. That is the direct question. This is the very easiest and direct question. Okay, this is logical questions. Okay, this is logical kind of question. So don't quit the question and with their keywords. Just read, understand. Okay, this is the apoptosis, I don't know this concept, so I move to the next question. That is not recommended, not right way. Whenever you are enter into this C part C, please you read the question at least twice. After the 2 time of reading you will not get the concept, just move to the next. That is the really appreciable, because that is not understandable to you. So don't waste the time in that question. So it's very important thing in the part C answering. Okay, next is dendritic cells, that is DC from BALBC mice. We are taken the dendritic cells from the BALBC mice, were treated with the interleukin 10 or with interferon gamma. Similarly, the dendritic cells from the B2 microglobulin in a deficiency mice were also treated with the interleukin 10 and interleukin gamma. So here they are taken the two type of mice. One mice is the BALB mice, another mice is beta 2 microglobulin deficient mice. So these two type of mice are treated with the interleukin 10 and R with the interferon gamma. After this treatment, they are taken the dendritic cells from the both mice. These cells were co-cultured with CD8 plus T cells from Ken-egg lysozyme. Okay, they are co-cultured with T cells from Ken-egg lysozyme specific T cell receptor transgenic mice. This is the, they are taken from a mice, transgenic mice, in presence of the HEL peptide that we all know, the HEL peptides. The 5 days later, CD8 plus T cells were assayed for target cell lysis. Which of the following combination will having a highest target cytotoxicity? This is the question is which is having the highest toxicity. As per their statement, we are all know the previously yesterday we discussed this same concept. Whenever you are adding the cells, combinating the cell culturing, co-culturing, the B cells are playing a vital role. Okay, that is Dendrix cells. In this target highly toxicity having cells are Dendrix cells. They are derived from VALBC, and that cells are already treated with interferon gamma, and it's additionally co-cultured with the CD8 plus cells. Why this concept? Why not the interleukin 10? In this, whenever you are studying that T that the T cells, okay, T cells receptors, the T cells are that having the uh, antibodies that against the own T cells, that interferon gamma cells, that are having the higher target types of toxicity. In this, another way of possibility is whenever you are taking a B cells that are shown a higher toxicity with the interleukin 10. So if they mention the T cells co-cultured with the T cells, so the output is interferon and uh, the CD8 plus cells. And one more thing, this BALBC is don't have any deficiency. In this microglobulin deficiency is here. Deficiency cells are not recommended to co-culture with the transgenic mice, because transgenic mice also the altered gene. So this is not recommended, that not toxicity, highest toxicity. Here highest target toxicity only produced in DCBALB bar C interferon gamma cells and CD8 plus cells. Next concept is a vascular endothelial catheter is an important cell adhesion molecule for endothelial cells. This is a statement. Vascular endothelial catheter is an important cell adhesion molecule for endothelial cells. Endothelial cells are that are unable to express VE, that is endothelial catheter, still can adhere to one another via N catheter, that is a neural catheter, but these do not survive. Which of the following statement is most appropriate reason for that? Okay, here also they mention what is the vascular endothelial catheter. So this is a cell adhesion molecule. This cell adhesion molecule, it's no, is expressed by some things, some cells. But here they provide the statement: endothelial cells are not able to express the VE. Catherine still can adhere to one another via N Catherine. So here this endothelial cells are not able to express the vascular endothelial Catherine, but that are make adhesion with the help of N Catherines. But these cells do not to survive. but attachment after that is only attached on the cell surface but not able to survive. Why? What is the reason for this appropriate? What is the appropriate reason? Sub B, the V, that is vascular endothelial catheter, act as a co-receptor for VEGF, that vascular endothelial growth factor mediated signal transduction in endothelial cells. So this vascular endothelial receptors are um, playing a vital role in a cell growth, but this N-cathedin is not helpful in a growth factor. That is the neural cathedin, not helpful for the growth. Okay, next very easiest question. In this question we want to read it again and again, then only we will get the correct answer. First statement: clearance of phagocytosed intracellular parasites. Clearance of phagocytosed intracellular parasite like Leishmania require the involvement of reactive oxygen species and reactive nitrogen species. Administration of interferon gamma to macrophage harboring an intracellular pathogen lead to production of the uh, reactive oxygen species and reactive nitrogen species by Jack and Stack pathway. Macrophage cell line J774 infected with Leishmania is given the following treatment. Okay, they are treating it with a interferon gamma plus AMT, a potent NOS inhibitor, and third one is treated that treatment is apocynin, interferon gamma plus apocynin. This is NADPH oxygen oxidase inhibitor. The next point is interferon gamma plus NMMA, that is the N-monomethyl L-arginine. It's arginine analog. These are four kind of treatments. After the infection, they are treating with the four-run way. What will be the most appropriate graph shown the survival of parasite after this treatment? So, after this treatment, that parasite want to be survive. What is the rate? Okay, this is if we are for example here there once again I read the question, sorry. The clearance of phagocytosed intracellular parasites like Leishmania require the involvement of reactive oxygen species and reactive nitrogen species. Okay, the statement explain: if we want to kill that Leishmania means that needs involvement of reactive oxygen species and reactive nitrogen species. So this is a clear statement. So if you want to kill it, we want this the ROS and RNS. Then administration of another statement they provided: interferon gamma to macrophages harboring the intracellular pathogen leads to production of the ROS and NS. Okay, so whenever you are adding the interferon gamma, that makes activation they are utilizing the ROS and RNS. Okay, in this way, macrophage cell lines J774 is infected with Leishmania. This J774 is just a one organism name. They are given this, they are not specifically mentioned it, just a microorganism. That is just a cell line. Okay, in this case, first is they are injected only the that interferon gamma, and then injected with the AMT, and then injected with the apocynin, and then injected with the AMA. Okay, here as per their statement, this cells they are having the interferon gamma, that interferon gamma releasing the ROS and they are releasing the RNS. This release lead to degradation of the cell. Okay, that kills the cells. Okay, in this cell degradation, it's helpful for the cell that calculate in the cell percentage. Here they, as per their statement, you can see that they are interferon gamma. If they are treated with the interferon gamma, the production of ROS and RNS is very low. Sorry, it's high in a cell, so the cell leishmania is degraded, that is clearance happens, it kills the that leishmanias. So this kill leishmanias are died, so survival of the parasites has become low. After this statement, you can see the ABC. Okay, that ABC is other things are treated with the additional compounds. That additional compounds are that not effective, that kills only 50% of cells. Okay, now you can understand, I think so. If you have any doubt, please raise in the chat box. Okay, next concept, ligand recognized two different cell surface receptors, that is A and B, on the same cell type. Receptor A, after binding with the ligand, is internalized along with the ligand, whereas receptor B, once again read, receptor A, after binding with the ligand, that is internalized along with the ligand, whereas B, so both are internalized. After binding with the ligand, activity of the intracellular domain, one particular disease is associated with the loss of receptor mediated signal transduction of ligands. Different observers inferred that the disease may be resulted due to the the four reasons that they provided. In that four reasons, which combination of our inference do you think appropriate for the disease? There are more reasons are there. The combination of two reasons is correct. Which one is means Q and R, that is loss of binding affinity of receptor B due to the mutation in the extracellular domain. Another case is a mutation in the tyrosine kinase domain, rendering the inactivity. It's inactivate. Okay, it's inactivate. So these reasons are the major reason. Another question, an important role of FAS and FAS and FAS ligand is to mediate the elimination of tumor by killer lymphocytes. In a study, 35 primary lung and colon cancers are there. Behalf of the tumor were found to have amplified and overexpressed a gene for a secreted protein. The secreted proteins that binds to FAS ligand. The main reason of survival of these tumor cells by this ligand binding protein may be attributed to decay receptor activity. Okay, this concept is very well known, well studied concept, decay receptor activity. This decay receptor cause activity, the FAS and VAX ligands are mediate the elimination of tumor cells by killer lymphocytes. Okay, it is a statement. The statement is it's eliminating the tumor cells by killer lymphocytes. And next statement, in the study, the 35 primer lung and colon tumor, they are taken the colon tumor. Half of the tumors were found to have an amplified and overexpressed gene that called the secreted protein that binds to a FAS ligand. Okay, that is bind to FAS ligand. The main reason of survival of tumor cells with this secreted FAS ligand binding protein is attributed to decay receptor activity. And these are all the concepts. Another important concepts, BALB mouse. This is another thing which is important in the part unit 4. This is the mouse was injected with that thymectomized thymectomized on the first day after the birth, whereas another's was thymectomized on the day 7. First mice is injected thymectomized on the day 1, and second mouse is thymectomized on day 7. Third mouse is underwent the same operation on 21st day. So first day, 7th day, and first mouse are treated at day 1, second mouse treated with day 7, third mouse treated with the 21st day. After 56th day, serum was prepared for the serum also from control mice. There is no treatment. There is no thymectomized mice. That no process happened. That is a normal BALBC mice. So there are 4 mouses are there which had the same operation. Say, and then serum was checked for anti-DNA antibody. Anti-DNA antibody. Which of the following observation is most pulsables? Okay, here in this concept, here they are having the big statements. In this statement, you want to read the statement and then understand the concept. While you are reading the statement, you will get understand. Okay, one is a control, another 3 are the treated. Here the answer, which one having the DNA antibody? Only one mouse having the DNA antibody. Why that having the anti-DNA antibody? In this case, that untreated, okay, that untreated mouse having the that anti-DNA antibodies. This that the concept called thymectomized. Okay, this in this case they are operated, and they are providing the removing the thymus. Okay, why they are removing the mice means they are in this thymectomized means that in this case they are treating the that in the treatment they are treating for the myasthenia gravis. Okay, in this surgery procedure, okay, we are removing the thymus gland. That if we are removing the thymus gland, there is no production of antibodies. So main things is the mice are treated. So in this concept, we want to know the concept called thymectomized. It's a very important key concept. Once you are understand, you are having the idea about this thymectomy, go to the your after the class, please refer thymectomy. This is the removal of thymus gland. After removing thymus gland, there is no production of the antibodies in the uh, human system. So without treatment, those mice which don't undergo the thymectomy alone able to produce the anti-DNA antibody. Next concept, a potential valuable therapeutic approach for killing tumor cells without affecting the normal cells in the use of my immunotoxins. Okay, question is a repeating one. In that question also they are asked. Here they are repeatedly asked the question, that concept called that antibody which carries the FAB region, it's not replaceable. We can conjugate these cells, we can't replace the FAB region. The keyword here is we can conjugate a compound or toxin with the FAB region of the antibody, monoclonal antibody. we can't replace the FAB region of antibody. It's a key thing, important thing. Next thing is the action potential was generated on a nerve fiber. If anyone missed this question previously, go to the back slides and refer this, okay, this concept. 59th, then 60th question, an action potential was generated on a nerve fiber by a threshold electric stimulus. This is the statement concept. When a second stimulus was applied, no matter how strong it was, during the absolute refractory period of action potential, the nerve fibers was unable to generate second action potential. This observation was explained in the following statement. Which statement is become right? The statement is true is R1, that is a large fraction of sodium channels was voltage inactivated. Their statement is, provided the statement, their action potential was generated on a nerve fibers by threshold electric stimulus was applied no matter how strong it was. During the absolute refractory period of action potential, the nerve fiber was unable to generate second action potential. Why it's not able to second action potential means a largest fraction of sodium channels was voltage inactivated. So this inactivation make a trouble. So there is no second, unable to generate second action potential. Okay, this is very easiest method. The following match the following. Okay, rabies. Okay, rabies. This okay first comes from the uh, Creutzfeldt's Jacob. This is a pathogen. This is caused by the pathogen called the prions. Okay, Creutzfeldt Jacobs call, it's made by the prions. Second concept, Q is pneumonocytes. It comes from the fungi. Lignoria's disease, it's caused by bacteria. Rabies is caused by the virus. This is a direct question. It's very easy question. Okay, next concept is I-25. I-25 labeled diaminofluorine. It's a well-known photoactivatable hydrophobic probe. The statement, it's a well-known photoactivatable hydrophobic probe of plasma membrane integral protein. Hydrophobic thing, very important. To determine the approximate length of number of hydrophobic chain domains in the integral membrane of protein, a control experiment, a following standard protocol is carried out in order to ascertain the foresight aspect indicates the correct combination of experimental protocol for the following choice. Okay, we want to select the following correct protocol. For the correct protocol is, first, intact membrane was allowed to interact with the DAF, that is the I-125 labeled diaminofluorine. Okay, first intact membrane was allowed to interact with the DAF. Then the membrane was solubilized with the detergent. We want to solubilize that membrane. Digested with the protease enzyme, that protease cleaving the particulars, run the STS page followed by the autoradiography. This is the sequential process. Okay, why this? The question they are asking, we want to digest. Okay, determine the approximate length and number of hydrophobic domain. We want to determine the approximate length and number of hydrophobic domains in any integral membranes. This is the statement. This is the question. For that question, what we want to do, intact membrane was allowed to interact with DAF. That is, DAF is a I-125 labeled diaminofluorine. This after this labeling, the membrane is solubilized with detergent for for the degradation of the membrane, disturbance of membrane. After that digested with the protease. The protease cleave the peptide bonds between all those membrane compounds. Then run the STS page to determine the size. And this is for the deduction, that is for the visualization, that is labeled with autoradiography. This is the step. In a plaque forming cell assay, antigen specific B cell numbers are assessed. This is the statement. In a plaque forming cell line assay, antigen specific B cell numbers are assessed. In this assay, antigen coated sheep red blood cells are lysed by the hepatine specific B cells by complement mediated cytotoxicity. It's another statement. In an assay that tried to enumerate the TNP specific B cells elicited in a TNP KLH primed cells. Plaques were formed despite the presence of antigen specific antibody presence of B cells. Which of the following is not the reason for absence of plaque? Here the absence of plaque is a reason is it's a complement having the anti-KLH antibody. If there is a presence of anti-KLH antibody, there is no plaque is present in the cell. Just read the question and then answer. Okay, we have some more questions. It's too late. Uh, I will send the people. Please follow this. Welcome to the upcoming day 15th class, tomorrow class. If you have any questions, please ask it in a telegram group, and you can ask it in a chat box. Thank you everyone. Now section.